The regulation could be exerted at transcriptional level, formation of primary transcript, processing level, regulation of splicing, transport of mRNA from nucleus to the cytoplasm, translational level. The germ in the cells are expressed to perform a particular function or the set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of disaccharide lactose into galactose and glucose. Bacteria use them as a source of energy. However, if the bacteria do not have electrons around them to utilize energy source, they would no longer require the synthesis of the enzyme beta-galactosidase. Therefore, in simple terms, it is the metabolic, physiological, and environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organisms are a result of the coordinated regulation of acceleration of several sets of genes. In prokaryotes, control of the rate of transcription and initiation is a predominant site for the control of gene expression. In transcription, the activity of the RNA polymerase at the given promoter is in turn regulated by interaction with accessory proteins, which affects its ability to recognize the starch. This regulatory protein can act both positively activator and negative repressor. The accessibility promoter region of the prokaryotic region in many cases regulated by the interaction of protein with sequence term operators. This operation region is adjacent to the promoter elements in most operon, and in most cases the sequence term operator bind a repressor protein. Each operator has a specific operator and specific respiration. For example, lac operator represents only in the lac operon and interacts specifically with the lac repressor only. The liquidation of the lac operon was also a result of the close association between the Johnson Francis Jacob and the biochemist Jacob Monod. They were the first to calculate the transcriptionally regulated system in lac operon. Their lac reported the lactose, a polycistronic structural gene regulated by a common promoter and regulatory. Such arrangement is very common in bacteria and is referred to as operon. To name such few examples: lac operon, trap operon, era operon, his operon, ball operon. The lac operon consists of the one regulatory gene. The I here term I does not refer to inducer data. It is derived from the word inhibitor and three structural genes ZYA. The I gene codes for the reparation of the lac operon. The Z gene codes for the beta-galactosidase, which is primarily responsible for the hydrolysis of the disaccharide lactose into monomeric units like lactose and glucose. The Y gene codes for permease, which increase permeability of the cells to beta-galactosidase. The A gene encodes the trans-acetyl, hence all the three gene products in the lac operon required for metabolism of lactose. In most other operons as well, the gene present in the operon are needed together to function in the same or related metabolic pathway. Lactose is the substrate for enzyme beta-galactose and is regular switching on and off of the operon has been induced in absence of preferred carbon source such as glucose. If lactose is provided in a growth medium of the bacteria, the lactose is transported into cells to action of permease. Remember, a very low level of expression of lac operon has to be present in the cell at all the time. Otherwise, lactose cannot enter the cell. The lactose then induces the operon in the following manner. The repression of the operon is synthesized all the time consequently from the I gene. The reparation protein binds to the operator region of the operon and prevents RNA polymerase to transcribing the operon. In presence of the inducer, lactose allows the reparation inhibited by the interaction with the inducer. This allows RNA polymerase access to promoter and transcription process. Essentially, regulation of lactose can also be visualized as regulation of enzyme synthesis by the subset. Glucose cannot act as inducer for lac operon. Regulation of lac operon regulation referred to as negative regulation. Lac operon and control of positive regulation as well, but it is beyond scope of this review. Translation. Translation refers to the process of polymerization of amino acids to form a polypeptide. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as peptide bond. Formation of a peptide bond requires energy. Therefore, in first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called the charging of tRNA. Amino acylation of tRNA to be more specific. If two such charged tRNAs are close enough, the formation of peptide bond between them would be favored energetically. The presence of the catalyst would enhance the rate of peptide bond formation. The cellular factor responsible for synthesizing protein is the ribosome. The ribosome consists of the structural RNA and about 80 different proteins. In its active it exists as two subunits, a large subunit and a small subunit. When a small subunit encounters an mRNA, the process of translation of mRNA to the protein begins. There are two sites in the large subunit for subsequent amino acid to bind to and thus be close enough to each other for the formation of the peptide bond. The ribosome also acts as a catalyst. 23S RNA in bacteria is the enzyme ribozyme for the formation of peptide bond, and translational unit mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide and are referred as untranslated regions. The UG are present at both 5 days and before the start codon and the 3 days and after stop codon. They are required for the efficient translation process. Third initiation, the ribosome binds to the mRNA at the start codon AUG that is recognized only by the initiator of the RNA. The ribosome proceeds to the elongation phase of protein synthesis. During this complex compound of an amino acid linked to tRNA and subsequently bind to the appropriate codon in mRNA by forming complementary base pair with tRNA anticodon. The ribosome moves from codon to codon along the mRNA amino acid added by one by and translated into polypeptide sequence dictated by the DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. Genetic code. During the application and transcription, nucleic acid was copied to form the another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transferring genetic information from the polymer nucleotide to form from polymer of amino acid. Neither does any complement exists between nucleotides and amino acids, nor could any non-theoretically. Therefore, existed ample evidence to support the notion that change in the nucleic acid genetic material was responsible for change in amino acid in protein. This led to proposition of the genetic code that could direct the sequence of the amino acid during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition deciphering of the genetic was most challenging. In a very solution, it is required involvement of scientists from the several disciplines: physics, organic chemists, biochemists, and genetics. It was George Gamow of physics who argued that since there are only 4 bases and they have to code for 20 amino acids, the code should be constituted a combination of bases. He suggested that in order to code for all 20 amino acids, they should be made up of 3 nucleotides. This was the very bold proposition because a permutation combination of 4-3 would generate 4 codon, generating many codon then required. Providing proof that codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with refined combination of bases, homopolymers, copolymers. Marshall needed a cell-free system for protein synthesis, which finally helped the code to be deciphered. Severa Okowa enzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequences in a template-independent manner, enzymatic synthesis of RNA. The salient features of genetic code are as follows. The codon is replaced 61 codon for amino acid, 3 code do not code for any amino acid, and they function as stop codon. Some amino acids are coded by more than one codon, hence the code is degenerate. The codon is rich in mRNA in continuous fashion. There are no punctuation. The code is nearly universal. For example, from bacteria to human, you would code for phenylalanine. Some exceptions to the rule have found in mitochondrial codon and in some protozoans. AUG has dual function. It is codes for methionine and also act as initiator codon. UAA, UAG, UG are stop terminator codons. The resistance gene adrenal burst and the mutation effects of large deletion and deletion in sequence of these are easy to compare. It may result in loss or gain of a gene or and so a function. The effect of point mutation will be explained here. Classical example of point mutation is the change of single base pair in the gene for beta globulin chain that results in sound of amino residues limited to valine. It results into a disease condition called the sickle cell anemia. Effect of point mutation is the release a base in structure gene can be understood by the following simple example. The consolidation propagation is very obvious. The insertion and deletion of one or two bases changes the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion or deletion. Insertion and deletion of the three or its mature base insert or deleted in one or multiple codon, hence once or multiple amino acid, and reading further remains unaltered from the point onwards. Types of RNA and the process of transcription. In bacteria, there are three major types of RNA: mRNA, messenger RNA, tRNA, and rRNA. All three RNAs are needed to synthesize the protein in a cell. The mRNA provides the template. tRNA being amino acid and reads the genetic code. tRNA plays structure and catalytic role during translation. These are a single DNA-dependent RNA polymer that catalyze transcription. All types of RNA in bacteria. RNA polymerase binds to promote and initiate transcription. It uses nucleoside triphosphates as substrate and polymerizes in a template-dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix and continued elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminated region, the nascent RNA falls up, so also the RNA polymerase. This results in termination of transcription. And the real question is that how is the RNA polymerase able to catalyze all the three steps which initial elongation and termination? The RNA polymerase is only capable of catalyzing the process of elongation. It associates the transcriptly with initiation factor and termination factor to initiate and terminate the transcription respectively. Association of these factors alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing to become active, and also since the transcription and translation take place in the same compartment, there is no separation of cytosol and nucleus in the bacteria. Many times the transcription can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. In equity, there are two additional complexities. There are at least three RNA polymerase in the nucleus. In addition to the RNA polymerase found in the organelle, there is a clear-cut division of labor. The RNA polymerase 1 transcribes RNAs 20S, 18S, 5.8S, whereas RNA polymerase 3 is responsible for transcription of tRNAs 5.S and nuclear RNA. The RNA polymerase transcribes the precursor mRNA, the heterogeneous nuclear RNA, hnRNA. There are at least three RNA polymerase division of labor. The RNA polymerase 1 transcribes three rRNAs 28S, 18S, 5.8S, whereas RNA polymerase 3 is responsible for transcription of tRNA 5S srRNA and snRNA, small nuclear RNAs. The RNA polymerase 2 transcribe a precursor of mRNA, heterogeneous nucleus RNA, hnRNA. The second complex is that the primary transcript contains both the exon and introns and are non-functional, as it is subjected to process called casing. When the introns are removed, exons are joined in a defined order. hnRNA undergo additional processing called as capping and tailing. In capping, an unusual nucleotide methyl guanosine triphosphate added to 5' end of hnRNA. In tailing, adenylate triresolute 200 to 300 are added at 3' and in a template independent manner. It is fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation. The signification of such complexity is now beginning to understand. The split gene arrangement represents probably an ancient feature of the genome. The presence of intros is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In recent times, the understanding of RNA independent processes in the living system have assumed more importance. Transcription unit and the core gene. A gene is defined as a functional unit of identity. Through there is ambiguity that the genes are located on a DNA. It is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for the tRNA or RNA molecule also defined a gene. However, by defined as a stone as a segment of DNA coding for a polypeptide, the structure, gene, and transcription could be said as monocystotic mostly in eukaryotes and polycystotic mostly in bacteria. Prokaryotes. In eukaryotes, the monocytonic structure gene have interrupted coding sequence. The gene in eukaryotes are split. The coding sequence are expressed sequences are defined as exons. Exons are said to be those sequences that appear in the mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of the genes in terms of DNA sequence. Interest of character also affected promoter and regulatory sequences of structural genes. However, sometimes the regulatory sequences are rudely defined. Regulatory genes to this sequence do not code for any RNA or protein.